Good day, everyone. How are you? Uh, the lecture today, before yesterday, we had was about mostly about the regulatory region of the gene and the control of gene expression. So today we are going to take a look at the coding region of the gene. So it's about the eukaryotic. It is related with, specifically related with the eukaryotic messenger RNA, like this, because the one that I'm going to talk about, the name is the introns, are only present in the eukaryotic messenger RNA, not in the ribosomal RNA, mostly, not in the transfer RNA, but mostly in the messenger RNA of the eukaryotic cells. Uh, Although, yes, some transfer RNA has a small section of the so-called introns, but we are going to ignore that for the sake of simplicity. So let's just assume that only this so-called some element called introns are only present in the eukaryotic messenger RNA. Let's just assume that. And another thing, a little bit of the complication is also, although the prokaryote, but the archaebacteria. Archaebacteria also do have the introns. So you cannot really say that these introns are only some luxury or some advanced feature of the eukaryote only. But anyway, so first, what are the introns? This figure illustrates well about the introns, the secret of the intron. So as you remember, these blocks area we can define as a coding or transcribed area of the gene, whereas this area, on the other hand, including the promoter, was the one we looked at it as the regulatory region of the gene. So both this regulatory region plus this transcribed region make up one functional unit of the gene. When you simply talk about the gene, you don't simply talk about this as only the gene, because the reason being is without this area, this, no matter how valuable information it contains actually in directly related with actually the final protein coding information, but still it's not going to be able to expressed. Okay? So without this regulatory region, the gene cannot be expressed. That's why you have to include those regulatory region containing the enhancers and promoters. And by the way, one question last time was about, you said actually the promoter is the only the binding site for the RNA polymerase. And then you also said that inactive gene versus active genes are the one that actually the transcription activator is the one bound to the promoter region. What's going on over here? Yeah, it's my bad, but also it's just a bad for all those scientists interchangeably using this term promoter. Specifically, to be more accurate, promoter, the term promoter should be used only strictly on those specific actual direct contact site for the RNA polymerase the enzyme. However, vaguely, when they refer all those regulatory area, a lot of scientists simply call these area as a promoter, because it's more difficult to specifically pinpoint, okay, promoter and enhancer and silencer, blah, blah, blah. Instead, simply put all those regulatory area as a, hey, promoter region or something. So that's the confusion that you get. Sometimes they specifically refer promoter as a very strict binding site for the RNA polymerase. Some other times, simply casually, promoter refers to the entire area of the regulatory region. Blame them, okay, including me. So that's the reason I said active gene versus inactive genes are the ones something that associated with the promoter region or anything. So anyway, so if you pay only your attention only to this coding or the transcribed area of the gene, now you immediately notice something. This is the case for the messenger RNA, right? Not the case for the ribosomal RNA or the transfer RNA, anything like that, but only pertains to the messenger RNA gene. What's the difference between messenger RNA and the others? Do you still remember? The difference between messenger RNA and the other all kind of different RNAs is what? Coding versus non-coding. Messenger RNA is the only RNA considered to be a coding RNA because it directly, once you have this, you can infer what kind of amino acid you will get out of this information of messenger RNA for the protein. However, all the other kind of RNA, no matter how hard you look at it, you will never be able to get any information about what kind of proteins because they are not involved directly in direct coding of protein. This is the only. Messenger RNA is the only one who is directly involved in actual the matching, the protein information. So anyway, we are going to take a look at that actual mechanism, how they link this information, the information to actual protein things later today, hopefully. If not, maybe next week only. So anyway, you immediately notice that this is called something primary RNA transcript. By the way, transcript means product of a transcription. Transcript. So transcript is a synonymous to what? RNA. Whenever you see transcript, that means in biology, it means RNAs, all kind of stuff. Because the process of making RNAs is called transcription, right? Actually, in your casual everyday routine life, the transcript means what? It's a copy. Transcript is a copy. When you apply for the foreign university, like for your graduate school or anything, you have to request your transcript at the registrar's office. Why are they called transcript? Your school record, including your GPA, is called a transcript. Why? Because your original record is so wholly original, so university will keep it. It's not going to release you the original. So all you can get is the copy of your record. So that's why it's called the transcript. And you send your transcript, which is a copy of your whole school record, to the prospective school. Then they decide, oh, this student is so good, so we are going to admit you or reject you, depending on your the previous record. That's the transcript. And actually, it's related with this. Why the products of the RNA, why RNA is called a transcript? Because it is actually a copy. Nothing but a copy of the DNA region. So this DNA region is simply being copied into this exactly. But when I say exactly copied, actually I'm wrong because it is not exact copying, but complementary copying. You have to always remember and understand that process. When there is a DNA sequence information, then the process of making RNA, which is a transcription, simply involves, oh, what is this particular nucleotide's complementary base? And bring it on. And the reason they can do this is, at least temporarily, DNA and RNA can hold the hybrid complementary base pairing. So by simply doing so, they can extend the RNA nucleotide assembling procedure according to the original information. That's why and how the RNA is being made in the form of this complementary copy is. Okay, so this. Do you see something weird thing? The primary means first the initial. When they were initially made directly out of this copying process, the length of this RNA, entire messenger RNA, is exactly equal to the area of the DNA that served as the template, right? Understandably so. But in the end, what they go through is this shrinkage process. And you can easily figure out what just has happened, right? Something were cut off, and then the remainders were joined together. As you see, right? You can understand. And those who cut off, read the label, all those removed, all those segments of the originally present, these are the segments originally present in the DNA as well, were removed. And they all, those who removed, had a common name, which are introns. Yes. Intron means simply intervening sequence. So this is useless junk that need to be removed, is some kind of implying their introns. So these guys, why do you originally in the first place are there? Because you were going to be removed anyhow. It's very funny, strange phenomena. So in the final project, actual mature messenger RNA, none of these introns are present. So only those survivors, the final survivors, the segment-wise, the final survivor, this, all had the same common names, which were exons, right? Only those exons remained, and all those exons, each originally, after the removal of intron, each exon segment present as a separate fragment, but they were all joined together. That's one of the jobs for those so-called the ribozymes involved. And this process precisely is called this intron removal and exon splicing, the procedure, or simply, or the entire process is sometimes casually called the splicing, simply splicing, RNA splicing. When you hear RNA splicing, that includes, usually originally include that the introns are removed and exons are joined together. That's entirely called RNA splicing. Right? Okay. So do all the messenger RNA of eukaryotes have these introns? No. Some messenger RNA of eukaryotes do not even have any introns. So they are more straightforward. So the entire coding region area of the DNA is exactly identical, equal in length to the final and the initial product of the messenger RNA. That's more, I like it. But most of the eukaryotic messenger RNA have this disrupting, intervening intron sequences that have to be removed during the process of RNA processing, so-called. This entire procedure is called RNA processing. And this processing occurs within the nucleus. Until it's finished, until it's accomplished, completed, this RNA cannot even escape the nucleus. So cell consider unprocessed this raw initial primary RNA transcript as an immature. So you're not, hey, where are you going? You're not going to be able to go anywhere. Stay there until I do something. So finish, trim all you, so that you look good now. So you're good to go. Then now finally, it can escape, it get out of the nucleus, and then in the cytoplasm, they can participate the process of a protein making, which is we call translation. Of course, there are two more additional trimming, editing procedure that need to be added onto before actually they get out of this thing, which later I briefly talk about that. So that's the nature of introns. Introns are being removed. As a gene, as a eukaryotic messenger RNA gene, if you have this intron in your DNA, unfortunately, those introns must be removed before this product of the gene expression, which we call transcript, do anything actually functionally useful. Okay, that's the thing. Then obviously you question why. You're going to remove this anyhow. Why do you have them in the first place? Because you have been living here for several billion years. Okay? Why didn't you? You had enough opportunity to get rid of them introns, because it seems that you don't need any introns anymore, right? No matter how, okay, maybe you can actually offer an excuse that, yeah, back then I was so primitive, so these are junk, useless segment of DNA had to be also included, carried in, but you had such enough long time of evolution. And you actually, your top priority is efficiency for your survival. And why didn't you get rid of them? You stupid. And then the answer is, hey, look at those prokaryotes, the bacteria. Bacteria cells don't have any introns. So it seems like actually bacteria are much more clever than eukaryotes, so that all those useless introns are long time ago, they somehow got rid of them. Only those a little bit more stupid archaebacteria still carried, didn't have a chance to get rid of this introns. So archaebacteria and eukaryotes still have this introns. I mean, this is like more inconvenience. Before you actually directly put your messenger RNA into action, you have to firstly remove all those introns, which is more costly and time consuming. As a eukaryote, it's a shame. Yes? Yes?
그러면 원시적이어서 이유 말고
Mm-hmm.
박테리아에서 이프론이 없을까? 원시적이어서 말고 그런 어떤 적응이라든지 환경 변화에 대한 적응 아니면 다양한 병변이 발생 정말 오히려 종족이 더 살아남기 관점으로
Everything. It's more useful for them to not have introns. For bacteria, having those introns removed is much more efficient for them for all these different purposes. Because their lifestyle is simpler, quicker. Okay? Maybe we have a subtle, indirect benefit coming from introns, but those introns benefit do not apply to the bacteria. So for bacteria, it would have been much more economical and efficient to remove those introns. But still, for some reason, eukaryote carry them. So then questions, why? So people don't know. There are two large groups, two schools of thought still arguing, hey, this is the origin of intron, and because of the origin, that's the reason why eukaryotes still cannot dump the intron. And the other school thought and argue otherwise. Yeah. So it's not actually, well, we don't have enough time to go over such their point. Simply put, scientists still do not exactly understand the reason why eukaryotes still carry the introns. But at least what we can say is, by having the intron, at least we can enjoy this benefit. It's called alternative splicing. So to give you one very good example of what am I going to do? What's going on? Jesus. I made a mistake something. Human. Before the beginning, before the start of the Human Genome Project, scientists have estimated that at least there's got to be at least the minimum 100,000 different genes should be present in the human, in each single human cell. Why? Because they already knew beforehand that the number of different proteins present in the human cell, which was about 100,000 different proteins out there. So you do the math. It's such a straightforward math. If there are 100,000 different proteins, then there got to be a matching, each equally matching 100,000 different messenger RNA didn't exist. And then it turned out, voila, you have, as a human, you have only freaking 22,000 such protein coding genes. What is going on? How do you manage it? You are not a Jesus Christ, so you cannot manage it with only 22,000 genes and then expand this into 100,000. What is going on? So everybody, including human, and it does not only apply to human, but to every eukaryote, plants, fungi, and all the other animals. They employ the same trick, also called this alternative splicing, which is simply this. You have potentially 1, 2, 3, 4, 5 different segments of exon, and in between everything else will be removed. But you can randomly, it's not really random, you can choose on your own. For example, you have 1, 2, 3, 4, 5 different segments that potentially that can become an exon, right? And instead of always every day using 1, 2, 3, 4, 5 as a primary transcript. And now, if 1, 2, 3, 4, 5s are always to become exons, then in between everything will always be introns. But that is not the case. Sometimes you generate this messenger RNA only having 1, 2, 3, and 5. So now, in this case, this segment 4 will become what? Intron. On the sunny days, maybe, this is a really gloomy, cloudy day. On sunny day, you may actually choose, oh, I want to pick 1, 2, and 4, 5. You can change your menu. This is what is called alternative splicing. So for a single gene, they can maximum employ this type of trick so that they can have 2 or 3 different versions of the RNA product by switching some of the segments of these introns. Then one gene can generate how many different proteins? Several. That's how they manage it. With only 22,000 genes, they can make more than 100,000 different proteins is the secret. And this is not, as I say, only humans' ability to display as a magic, but everybody else can do this. They do that. So that's at least one obvious advantage, isn't it? You can economize using only compact genome. You can expand much more than your actual genome is capable of. You can expand the repertoire of the proteins. So this is a real example in that one such gene, by switching this segment as to become an exon or intron, simply you can change the destiny of a cell. So by having this, then this protein can be inserted on the cell membrane, then this cell will be doomed to death, cell suicide. It will kill itself. And on the other hand, sometimes then by excluding this part, then this protein will stay in the cytoplasm, stay there, do not go into the cell membrane, then this cell will be spared. This cell will survive type of a different switch. So if you can extend this thing to your imagination so that different type of cells in different location of your body can employ this trick so that they can have different version of proteins. Or in terms of a timely different protein can also be produced like this. So at your early stage, when you're a baby, you can produce this particular version of a protein, and later on, you don't need to make up another gene for that. Simply by switching the little segment of the intron, the type of a protein produced at adulthood, adulthood will be totally different. Okay? Yes?
그러면 그 아까 문제에서 보여주 PPT에서 보여주신 것처럼 슬라이드에 따라 전혀 다른 것이 나오는데
Yes.
어떻게 유전자의 그런 mRNA 다른 결과를 다르게 하는 것은 무엇이고 또 그런 다르게 하기 때문에 그 투명 생태가 얻는 이점은 뭘까요?
The differential expression, gene expression thing was explained in the last lecture like that. So the differences in gene expression in terms of different amount or different type of genes control is controlled by transcription factors, right? On the promoter region. But once they are done and you have another second level of additional level of this control in the messenger RNA level, this has already beyond the expression. They are all expressed. And after the expression, this messenger RNA version can be different. It's two different levels of the expression, the control, gene control is there. Confusing?
그러면 나이에 따라서 달라지는
Oh yes, of course, yes. There is actually examples of the different age of these things. Excuse this mess. So that's about the alternative splicing and the contribution of this alternative splicing to the actual genetic variety, diversity, is what we can have verdict. Now, jumping into the actual protein making process, which is called translation. And once I mentioned that why this thing is better be called translation, what it means by why this is particularly referred as translation. Because up until now, up until the level of the messenger RNA, this is all about simply information, conceptual thing. The messenger RNA cannot do anything. Of course, some messenger RNA can act as an enzyme, so it has some functionality. But other than that, messenger RNA or DNAs are all simply just talking. You are simply talking the concept, information. Now, talking is good, great. Concept, the philosophy is great, but actually, can you provide us some bread? Is what the protein can give us. So how simply juggling this information only, how do you actually transfer this information into actual products of a protein? So that's why it is called the translation. This information, messenger RNA information, the information about instruction of how to make protein is written on the messenger RNA as some kind of genetic code. And not everybody can understand that information. In the cell, there are only one particular structure called ribosomes. Ribosome is the one who can only read this information and understand, oh, I see what you mean. Okay? When something is written in code, then ribosome understand, and then actually they can follow this instruction and then build. How do you make protein? Actually, without cell, if not cell, then in the test tube or on the laboratory bench, is there any way you can synthesize protein? There is? Yes, there is. You can synthesize protein simply by employing chemical reaction, the peptide bond reaction artificially, so you can assemble individual amino acid into protein, but that would be extremely inefficient and expensive. Yeah. So you cannot function like that. Although you can try, then you can do something, but not even any protein product for drug can be produced like that because it's too expensive and inefficient. This cells machinery by which these proteins are being synthesized is so marvelously, amazingly efficient. So anyway, so then we have to understand this code. Genetic code is being converted into actual the sequence of the amino acid. The reason why I brought up this the example of a machine, protein synthesis machine, is whether you are building the protein in the laboratory bench or in the cell by ribosome. Actually, all you gotta do is simply assembling each different individual amino acid in specific order and connect them. Connect each different amino acid by through specific chemical bond. The names, if you are curious, the name is a peptide bond, but who cares? The names is simply. Okay, so what is codon? Three sets of. So sets of three nucleotides present on messenger RNA. If those were present on the messenger RNA, obviously this actual exact same codon also should be present on the DNA too, because RNA, messenger RNA is, after all, exact copy, or not the exact copy, but complementary copy of the DNA. So this codon, the three nucleotide present on the DNA as well, on the messenger RNA too. And why this codon is so important? Because using these three nucleotides, just like as if we are using three alphabet to designate, to make up a one single vocabulary word. This using random combination of three nucleotide in order, you specifically make this three nucleotide mean something, just which is in this case the amino acid. Well, that's the concept of a codon. So for example, if you do this, then there are, because nucleotide consists of only four different nucleotides, so there will be 64 different ways of generating different types of three nucleotide set. Okay? 64. So for example, the reason why you see U is we are assuming this is in the presence of messenger RNA, so it's gotta be an RNA nucleotide, so U instead of a T. But if that was on the DNA, it should be ATG. So AUG, MET, what is MET? New York MET? No, it's not methionine. Usually we call this amino acid as a three-letter naming system we employ usually. In some occasions, some more professional users prefer to have a one-letter designation system, but anyway. This AUG signifies methionine, GGG, glycine, CCC, proline, type of those things. Okay. Don't try at home trying to memorize this. That's very dangerous. Don't do that. So it's useless if you try to memorize all this genetic code. For what? So yes.
교수님, 근데 진정세균들의 경우 A
Formulate,
메사인의
formyl group is attached to the methionine. Ignore this. You're not going to be able to having to use this. But the methionine, yes, in prokaryote, the bacterial methionine, the first methionine contains the additional chemical group, formulated group. But that's so anyway. So let's start with the template DNA. Your template DNA sequence is this. And your template DNA sequence is double-stranded DNA, so it should be its partner DNA. So together, this will make up a double-strand DNA sequence, right? But as a messenger RNA, you are only trying to copy one of the two DNA strand, and then the one you are copying in the form of a complementary base pairing is called the template DNA. And as you can easily notice that messenger RNA sequence will be complementary to this template DNA sequence in that T, A, G, C, A, U, like this complementary thing will always go as a rule. But once you also immediately what you can notice is the complementary sequence of the template DNA, this DNA is strangely called sense DNA. Why is it called sense DNA? Then you can easily realize that if you compare this messenger RNA sequence and the sense DNA sequence, actually it's more informative for the messenger RNA sequence because they always have this kind of complementary base pairing relationship. So if you are really curious about the messenger RNA sequence itself, then you look up the other strand of the DNA, then it will be exactly identical, except for this T. Hey, go away, go away. This T to U conversion, which is obviously the case because of the DNA and RNA nucleotide differences. So that's what is called the messenger RNA sequence. And this three, once it becomes a part of messenger RNA, then this information, the sequence, is read in triplicate. You read this one, ACU, CCU, GAC, in this way. It goes around until the end of this messenger RNA. And what's the point? What's the big deal of this codon? Because this codon, each specific codon, as I said, will specify specific amino acids like threonine, proline, glutamate, lysine, just like that. And the question is how? Because of another complementary relationship between codon and anticodon. If you imagine, ACU, this codon, is complementary to this particular sequence UCA. Where did it come from, this UCA sequence? What do you think? This particular UCA sequence is being provided by who? Guess who? Transfer RNA. Transfer RNA is an RNA too. As an RNA, it can also engage in the complementary base pairing. So if transfer RNA has a specific area site that exactly matches with this particular codon, then they can bind. They can join together. So just like messenger RNA can join to particular area of the DNA through this complementarity, that's why a certain segment of the DNA can serve as a template DNA. And you generate the messenger RNA. Messenger RNA have these sequences. And you have, there is a reason why we have a certain, obviously, all of a sudden, we have some kind of rule that you have to read, from now on the messenger RNA sequence, you have to read by threes. There is a reason why. Because this 3 unit will become a partner of some another sequence, mysterious named sequence called anticodon sequence. And if you examine, yeah, exactly that is the case. ACU, UCA, which is complementary. So these three complementary nucleotide sequence are the part of individual tRNA. Get it? Because what the tRNA does? tRNA, different kind of tRNA will bring each different amino acid. tRNA's function is what? Carrier of amino acid. Different types of tRNA can attach to different type of amino acid, and then they can come to this protein synthesis site, which is ribosome. And how they can have any kind of specific relationship while they have this specific complementary base pairing capability, they can bind between this part of the messenger RNA and this particular tRNA. Still a little bit fuzzy. Let's keep going. We'll be able to see a better picture about this thing later on this lecture. So anyway, as I mentioned, there are 64 different such codon can be generated in this way, but the funny thing is you have only 20 amino acid available in the cell. If you had a 64 amino acid, that would have been great, so that you should be able to offer much more variety of different kind of proteins, but unfortunately you have only 20 different amino acids. It's not that actually there are only 20 different kind of amino acids present in the whole world of chemistry. No, it's not. Probably there are even exceeding hundreds different type of amino acid possible exist. But for some reason, there are only such, among those different kind of amino acid, only these 20 amino acids are the one that become a part of the life's proteins, wherever you look at it. So actually, some crazy scientists have attempted to create new additional codon and create new additional tRNA that can attach to a different kind of amino acids, so that we can expand this repertoire of the amino acid, so that we can expand the variety of different proteins they have attempted about several years ago, and then they have succeeded in bacteria. So even such kind of crazy attempt have been successfully made, but other than that, there are only 20 different amino acids are there. So that means this vocabulary, redundancy, because there are 64 and 20. So obviously one amino acid will be able to handle more than one codon. So 3 or 4 different codons will specify the same amino acid. It's called the codon redundancy. Because of that. And because of this redundancy, some of you may think that, hey, it's in the first place, it has been just a waste. Why didn't you? Why did you choose, stupid enoughly, choose to use a 3-letter codon, so that expand this possibility of this different combination into 64. If you reduce them, somehow you could reduce this possibility to smaller numbers so that you can economize this thing. But look how. If you reduce this combination thing, the codon thing is from 3 to 2, then suddenly your total number of different amino acids that can code using that is reduced to 16, which is not enough. You have to have at least 20. So 3 was the reason. There was a reason why cells still had to keep this 3 nucleotide codon thing. Otherwise, they could not cover at least those 20 different amino acids to have these codon things. So now this anticodon, I try to emphasize the anticodon, but your look, most of you didn't really look buying into understand, okay, what's exactly the codon is. If the codon was the 3 nucleotide present on the messenger RNA, then the anticodon is such matching complementary nucleotide present on the other end on the tRNA is the concept. So that's because of such relationship, specific part of the messenger RNA should always match to specific type of a transfer RNA. And that's how you arrange different order of amino acid according to the different codon sequence. If different codon, remember codons are some specific 3 nucleotide on the messenger RNA. And if this codon 3 nucleotide has a complementary base pairing with transfer RNA sequence, and according to different codon sequence, then different types of transfer RNA can be aligned, arranged in this order. And different transfer RNA means a different type of amino acid sequences. And then all you gotta do is, while they are available, while just 2 specific transfer RNA are on same site together in the neighborhood, all you gotta do is simply quickly connect these 2 amino acids, because all these each different transfer RNA came in carrying specific their respective partner amino acid. They are not allowed. Without the amino acid, this transfer RNA is not even allowed to enter the site of the protein synthesis. That's the secret. And that's how you go through, proceed the procedure of a process of translation. What in English? So this is the 3D structure of tRNA overall. So it's all made of individual nucleotide as an RNA. tRNA is an RNA. And if you look closely, at one end here, at one end of tRNA is actually the one you see. What you see is a particular amino acid. Amino acid is attached. So this particular tRNA is keep cycling after hooking up particular its specific partner amino acid, then go into the protein synthesis site, which is a ribosome, and then it loses its amino acid during the process of protein synthesis, and then it escapes, and then go out there, and then hook up another, the same kind of amino acid, and come again when there is, but only this tRNA can come again only if what? Only if it can have complementary base pair with specific codon 3 sequence. And where is the place of such a specific complementary base pairing place? Here. The other end of a tRNA, there is a 3 nucleotide specifically, right? This is what we call anticodon sequence. 1, 2, 3 anticodon sequence, which can pair with specific messenger RNA codon sequence. And then they can hang on by virtue of this hydrogen bond of this complementary base pair. What's the reaction? So everybody's okay with my explanation? So verbal explanation of this is this. Each tRNA carries a single anticodon, the type of amino acid carried by this is determined accordingly, blah, 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 and whatever. Big deal. So that's just exactly what I've been trying to explain. Now, I said there are 64 different codons are possible. If you simply randomly, 4 out of 3, you pick up. How many different ways you can pick 3 different sequence? 64. But in reality, only then it means each codon should match with each anticodon, right? Each 3 codon should have theoretically matching anticodon sequence, okay? So it means there's got to be a 64 different kind of transfer RNA also should exist in the cell. But life is not fair. Yeah, there's got to be a 64 different, because there are 64 different codon. So if this thing is going to work smoothly, and there's got to be a exactly matching partner, 64 different type of anticodon, which means each different anticodon should be carried by different kind of tRNA. So there should be 64 different types of tRNA exist in the cell. But in fact, no, there is only 61 tRNAs. What am I going to do with these three missing codon? Then actually it turned out to be a blessing than a disaster. These three missing codon now can serve as a stopping. Enough is enough, you stop there, codon. And that's why it is called stop codon. Now, this is the codon table. Actually, to figure out what exactly combination of this particular codon actually matches to what kind of different amino acid has been really heck of a job, must have been, right? And one of the clever scientists finally figured this thing out and through elaborate experiment. And obviously, he won, obviously, deservingly so. He won the Nobel Prize. But anyway, thanks to that, we enjoy this information in these days as part of the codon table. So as you look at it, I mentioned that codon redundancy, right? So all these four different codon, UCU, UCC, UCA, UCG, yeah, they all code the same amino acid, the serine, just like that. So in this way, 64, the abundant and redundant codon can be allocated into different 20 amino acids. But notably, you can notice is these three combination of codon, UAA, UAG, UGA, those three are the one used as something like a signifying stop of this translation procedure. Why? And if you think about the reason why these three codons are used as a stop codon, is simply because for these three codons, there are no matching tRNA anticodon. So if you don't have any tRNA, no incoming tRNA can bind to this site. So this ribosome cannot proceed one step further. Still waiting. Where is this is empty. Where is this tRNA is going to come? But there is none. If ribosome was so patient in personality, then probably keep waiting for 5,000 years or something like that. But ribosome usually is not that patient. So like waiting for maximum couple of minutes, not even a couple of minutes, I guess, maybe several seconds. Is that coming? Okay. I had enough. I'm going home. And disassemble. That's how the translation stops. So without this stop process, then once the RNA is being made, it will go all the way down and it will become a chaos. So thanks to the presence of the stop codon, a certain messenger RNA can have some kind of defined length is the one. So figuratively, it can be explained like this. Although there are more well-drawn, more sophisticated figures you can easily find on the internet and or anywhere else in other types of biology textbook, but this is one of those translation explaining figure found in one type of simple general biology textbook here. So translation start. So it's described, illustrated in the middle of this procedure. One thing is there are two rooms, not exactly two, but there are actually three, but let's just simplify that. Let's assume that there are two spaces available for each time in the ribosome. The space for incoming transfer RNA. So in other words, in a given time, there are only maximum two transfer RNA can occupy both sites. like this. So when this, how a specific particular transfer RNA can come in is determined by what? If I may ask you, a specific tRNA in a given time point during the process of a messenger translation is going to be determined by what? Who determines who is going to be invited at this? So, like, let's say this is a transfer RNA A and this is transfer RNA B. How come this A and B can come to at this particular point on this site? It's going to be dictated by what? This messenger RNA sequence. The messenger RNA codon sequence will invite the partner anticodon. The transfer RNA having exact anticodon sequence will be able to come. So in other words, this sequence and this sequence will be what? Complementary. Please understand? And again, this particular codon sequence and this sequence of the transfer RNA will be complementary. But if this ribosome move one step ahead, this ribosome is moving this direction. If this ribosome go one step forward, and this amino acid, this transfer RNA will be kicked out. This amino acid will be kicked out, and this amino acid will move to this site. Now make this empty seat available. And this empty seat was actually, messenger RNA sequence was having this particular codon sequence. Now, what kind of transfer RNA will come will be exactly determined by what kind of a codon sequence is there. So in this way, this messenger RNA sequence, as this ribosome go along all the way down to, starting from 5' of this messenger RNA toward the end, the 3' end of messenger RNA, it will be always dictated by the specific sequence of this codon, 3 nucleotide. And according to that, what kind of transfer RNA will come and go will be exactly determined. And by doing so, each individual, why the older transfer RNA has so many amino acids attached, and then this newcomer, new incoming transfer RNA has only one. This is a norm. This is a normal status of any given transfer RNA, having only one single amino acid. But the reason why this old transfer RNA has several amino acids already attached is because of this. It has gone through this transfer procedure. Whenever these two amino acids are together, amino acids will be delivered from one transfer RNA to the other, and it will keep adding on just like that. So eventually you have build up long chain of such amino acid until what? When does it stop? Until this ribosome encounter with this particular sequence of codon, which is either one of those three what? Stop codon. Why you stop there? Because now there is no transfer RNA can come into and occupy to have this complementary base thing. So, this is the end point now. The ribosome will discharge, disassemble, and all those translation procedure will be over. I don't know. So that is, to some, it's just some easy, straightforward process to understand. And for some others, those who do not have any previous background information, must be very hard to grasp at first hand but simply it's just a matter of time. A lot of times, it's just a matter of time how many times you try to expose yourself to particular information, then overall, without knowingly, unknowingly, you get used to it, and voila, naturally, you realize that, oh, now I know it, is probably the only advice I can offer. So this anti-codon sequence explanation, including this stop codon, is there for you to read your extra time. Okay, so for the regular lecture, I will stop here. And then for the remaining 15 minutes, I will turn into your student question once again. Today, I didn't upload the previous student question because there was some unfinished business in the previous sector. So let's take care of it first. And what I'm saying is I really, really admire your different questions. So no matter how much more unnecessary time I have to spend extra, I really think this is like more beneficial for you to have this course. So I'm going to keep taking care of these questions. So this question is about why there are, among those 6 different kind of RNA that I introduced during the lecture, for the messenger RNA, ribosomal RNA, transfer RNA have been relatively found earlier, but then this particular messenger, not SN, SNO, and microRNA discovered so later. Or actually, the ribosomal RNA and the messenger RNA have been discovered kind of accidentally, and they are much more abundant. Ribosomal RNA and messenger RNAs are much more abundant than these RNAs, and their functions are much more general and much more akin to the quest for the life questions. Then these were the later, when they figured out the basic scheme of a central dogma, and then they look around, okay, what else are there? And that's how they come up with this. That's why these different types of RNA have been discovered relatively much later than this. And as I earlier briefly mentioned on the previous lecture, that there are actually one such example is the of RNA and DNA natural hybrid is during the process of the transcription. During transcription, a part of DNA is always having the hybrid complementary base pair with messenger RNA. So that's what. Another example is this so famous in these days, the CRISPR, that genome editing tool. This CRISPR machinery is actually a defense mechanism employed by bacteria against the viruses, bacteriophage. And the way they exert this effect is by having temporarily this hybrid between RNA. This yellow line is an RNA, and blue line is DNA. So when they have this particular hybrid complementary base pair, and this will be, as you can see in this scissor cell, this whole big ugly thing is a protein, just like a risk-like complex in the case of microRNA. This CRISPR thing also has this protein. It's called the Cas. Feels like drinking beer, right? So this will cut off the bacterial DNA sequences. So this is a part of their bacteria prokaryotes immune system. They remember By virtue of this complementary base thing, they remember what kind of viruses actually have actually entered. And then when those viruses come again, and you are going to be cut off miserably. It's just the CRISPR mechanism. But clever scientists found a way to utilize this thing in our advantage, and now we use this in editing our genomes. That's why it earned even the Nobel Prize very quite recently. Uh, in gene therapy, how is a specific gene turned into an activated state is uh, it, in many different occasions, it's just a case by case, so to speak. So I cannot really say in one very specific way, because in some cases, when you employ this particular method of gene therapy, it's because sometimes it's due to a specific gene is entirely missing or inactive, and you simply by providing the new copy, functional copy of this gene to this organism, individual in these cases, and then try to cure this defect. And some other times, actually, inactive gene turning into active one. If there was specific case, then you have to handle some expression level by trying to correct the promoter region. Technically speaking, if you try to employ this gene therapy method to not an embryonic stage, but already one individual like adult or even child, then it's technically become more difficult, because by treating one particular type of tissue in this method does not guarantee that all this method will spread all over the other body, usually, if you think about it. And in that case, actually, people have just come around, although it is not really entirely 100% successful, but like a virus, infecting virus, DNA viruses, you employ the disarmed, unharmed, harmful virus and let them spread all over the body and try to cure those things has been one of the main approaches. What do you think? If we had a reverse transcription like transcription, uh, you have to understand what would produce, what would this reverse transcription mechanism produce in the cell. This reverse transcription will produce what? DNA. You have so many RNAs. Reverse transcription will pick RNA as a template and copy this RNA information into DNA. That's what this virus's job, some particular types of virus. If we had, as a human cell, for example, if we had this functionality, although we do have a very similar reverse transcriptase-like function in at every end of the chromosomes, but that's a little bit different, so I'm not going to mention. But if we simply imagine what would the scenario that we had this as a normal function, the reverse transcription function, then we would create so many different types of DNA out of RNA. Would it be really something that we want? No. So that's why all those, the eukaryotic cell, including us, ourselves, don't need any reverse transcript, the enzyme capable of this reverse transcription. It will create so many different types of DNA in the cell around. DNA's place should be like confined in the nucleus. Okay. It's not really desirable for DNA to hang around everywhere in the cell, including cytoplasm. That's all just a mess. Yeah. This one, as I mentioned earlier, this prion protein is something is not related, directly related with the central dogma thing, because this, as a protein itself, is not acting as a genetic material. It's something behave as if it is an infectious agent. So earlier, scientists, some group of scientists believe that actually prion protein is a type of a viruses, but it turned out that no, it is not virus, but simply protein, very mysterious protein, which is capable of switching, turning normal protein into their mutated. This prion protein is a mutated protein, just like zombie. And this zombie, just like by having, grabbing and biting the so-called normal human and turning them into zombie, this prion protein do the exactly same thing by grabbing normal protein and then turning this protein into the structure of this mutant protein. That's it. This one was this microRNA thing was actually, it was discovered in this particular worm called the C. elegans worm very, very long time ago, relatively speaking, by these two scientists. But this was like beginning of the realized the mechanism of microRNA. But actually it began as they realized this RNA double strand things is being employed as some kind of defense mechanism for incoming infecting viruses was the one. And then finally, only this year, this work, because this work has been the pioneering work for discovery and an understanding of all those different microRNA work mechanism. And now finally, they received their long due, overdue credit. That's how.
아니 근데 실제로 술 같은 데에서는
That's their
mRNA를
job. I know.
mRNA라고 했잖아요.
You know how many stupid reporters are out there. They don't understand the difference between micro and mRNA.
이거 완전히 다른가요?
What can I say? Do you know the other thing around? How do you pronounce this? Famous basketball player. All those reporters in Korea, all those reporters pronounce him as a LeBron James. LeBron. No, it's not. It's called LeBron James. It's LeBron James. Everybody calls him LeBron James, but only in this country, reporters always write LeBron as LeBron. So what? Find out, and they will learn, but they never find out. Okay, I will stop over here today's lecture. Have a nice weekend.
그러니까 언론에서 그 진짜 엠브로스 박사와 제리 러브콘이 노벨 생리학상 받았었을 때 mRNA를 mRNA라고
Korea,
했거든요.
I know.
알아요?